Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokde from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibaut Van Zelle from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN-Topical Research Symposium.
I think the next speaker is Dr. Jozefien Declercq, who is one of the residents at Ghent University Hospital at the ENT department, and she is also doing a PhD in our lab and also at the VIB, and she has been working and doing a lot of, let's say, clinical and research work during the COVID pandemic, and she will present, I think, some results about her work.
Okay, thank you for the introduction and the opportunity to present our data here today. I would like to tell something about the mucosal humoral responses are induced in the nasal cavity after SARS-CoV-2 vaccination. So it's now almost 2 years ago that global vaccination programs against SARS-CoV-2 infections started, but already quite short after these after the start of the vaccinations, there were reports coming in that the currently used SARS-CoV-2 vaccines protect very good on the long term against mortality and severe disease, as you can see here in the lower graph. But the vaccine effectiveness against infection by SARS-CoV-2 is declining already 2 to 3, 4 months after vaccination. These are figures from one of the first reports, but currently many reports have been claiming that the long-term protection against SARS-CoV-2 infection from the currently used vaccines is rather poor. So we were wondering how we could explain these observations. And to answer this question, we believe that we need to go back to where it all starts, so the upper airways, because this is the entry of the virus, and here the virus replicates locally, and from here the virus can then go to the lower airways and possibly systemically. If virus neutralizing antibodies would be present in the upper airways, the virus would not be able to enter the cells, and consequently there would be no virus replication and no infection by the virus. And this has also been shown in experimental mouse models where virus neutralizing antibodies are intranasally administered, and these mice were protected against infection when they were exposed to the SARS-CoV-2 virus. So the burning question is now, can COVID-19 vaccination induce SARS-CoV-2 neutralizing antibodies? And to answer this question, we collected serum samples and nasal secretions from patients before and after vaccination. And then we ran a surrogate virus neutralization test on those samples. The first results we have already published. Here we had a cohort of 46 study participants, of which half were vaccinated with the Pfizer vaccine, so the BNT162b2, and half were vaccinated with the ChAdOx1 or the AstraZeneca vaccine. And we sampled those patients just prior to their first vaccine and sampled them again 2 to 4 weeks after their second dose of the same vaccine. And then we found that around 96% of patients that were vaccinated with Pfizer vaccine had nasal neutralizing antibodies, while only 60% of the ChAdOx1 vaccinated individuals had nasal neutralizing antibodies. And the induction of those local antibodies was irrespective of prior COVID-19. So here you can see in the third graph that the distribution of patients, of study participants that had nasal neutralizing antibodies is the same in the group that did have prior COVID-19 and those who didn't. So now that we know that those vaccines can induce nasal neutralizing antibodies, we asked ourselves if how long these antibodies are staying there in the nasal cavity, and if they could maybe explain the waning vaccine effectiveness against infection. And herefore, we extended our cohort to 183 study participants that were sampled just prior to their first vaccination, sampled again 2 to 4 weeks after vaccination. And then we followed them up until 6 months after initial vaccination. And we sampled our cohort again after they received the booster vaccination. And again, we followed them up for 6 months after booster vaccination. And then here you can see all the results we obtained. And you nicely see that in both the ChAdOx1 and the Pfizer vaccinated patients, there are nicely neutralizing antibody responses induced in the serum. They decline over time, but they stay present in both groups. And booster vaccination increases the nasal neutralizing antibody level, the systemic neutralizing antibody levels again. And these levels after booster are maintained for a longer time than just after initial vaccination. When we then take a look at the local humoral responses, you see that the results from our initial cohort were confirmed, that the majority of patients vaccinated with the BNT162b2 vaccine had nasal neutralizing antibodies, while in the ChAdOx1 group, only half of the patients had nasal neutralizing antibodies. And the very interesting thing is that we see 6 months after vaccination that those nasal neutralizing antibodies are in the majority of the patients not present anymore. As you can see, the dotted line is where we still see below this line, there is no virus neutralization. And you see that the majority of patients are under this dotted line. But the hopeful news is that booster vaccination increases those nasal neutralizing antibody levels again. And the initial differences between the Pfizer and ChAdOx1 vaccinated participants is gone. And 6 months post-booster, the majority of the patients still has nasal neutralizing antibodies. So it seems that booster vaccination can induce a more robust local humoral response. However, when you take a look at the lower graphs, you can see that there is quite some heterogeneity between all our study participants. And currently we are trying to understand why some patients retain high nasal neutralizing levels and while others don't have nasal neutralizing antibodies anymore at 6 months post-booster. So here you can see then the correlation between the serum neutralizing antibodies and the nasal neutralizing antibodies. And you see that at each time point, they correlate very nicely. So the systemic humoral response correlates very well with the local neutralizing response. And this suggests maybe that it's possible that antibodies from the serum might exudate through the vessels in the tissue. However, we're not sure about it, and it's something that we are currently further looking into. And one of the things that we did to answer this question is to determine the isotypes of the virus neutralizing antibodies. We performed an ELISA for anti-receptor binding domain IgG and IgA antibodies. And if you look here, you see the results from the from the IgG ELISA, and you see that both in serum and nasal secretions, IgG antibodies are induced in both the BNT162b2 and the ChAdOx1 vaccinated individuals, and they follow more or less the same kinetics as the neutralizing antibodies. And then if you look at the results of our IgA ELISA, you can nicely see that IgA is induced in both serum and in nasal secretions, and in both participants vaccinated with BNT162b2 and ChAdOx1. So it seems that both antibodies are induced in serum and nasal secretions, so we still don't have an argument that there is local production. But we looked at the levels of IgE relatively to IgG, and then you see that these relative IgA levels are higher in the nasal secretions compared to the serum. And this is even more pronounced in the ChAdOx1 group compared to the BNT162b2 group. So it seems that there are more IgAs in the nasal cavity compared to the serum. And that might point towards possibly local production in the tissue of SARS-CoV-2 neutralizing antibodies. But this is still something that we are further looking into, and we hope to have a definitive answer. Okay, to conclude, we know that SARS-CoV-2 vaccination can induce nasal neutralizing antibodies, and that those antibodies wane around 6 months post-vaccination, which might potentially explain the waning vaccine effectiveness against infection. So it seems that those waning levels of nasal neutralizing antibodies might be a biomarker of waning vaccine effectiveness, and based on this data, we advocate for the inclusion of those measurements in vaccine efficacy trials. Moreover, we see that the nasal neutralizing antibodies remain higher after booster vaccination. However, we see quite some heterogeneity between patients, and the question is now who remains high and who remains low. And we know that both IgA and IgG are induced upon SARS-CoV-2 vaccination, but there are differences between serum and nasal secretions, and we are currently looking into whether it's really a local production of these nasal neutralizing antibodies. And I would like to thank all the people involved in this project. Professor Philippe Gevaert, who is the main PI of this study, also Nathalie de Reuk, who did a lot of the practical work involved, and Steen van Hee, who is always ready for ideas for this study. Thank you very much for your attention, and I'm happy to take any questions.